Our research is in cancer genomics. So our largest focus is on what we call molecular archaeology of cancer. So we start from the genome of cancer cells and try to infer the tumor's evolutionary history from whole genome sequencing data. Basically, we try to reconstruct the evolutionary history of cancer from its whole genome sequencing data. So that is, in a way, looking at the cancer's past, or looking at the tumor genome as an archaeological record of the tumor's past. So that's why we call our approach molecular archaeology of cancer. It's important to understand as much as possible as we can about the tumor's evolution and how tumors evolve, what the early steps are, what the late steps are, such that we can then try to interfere with those steps optimally and in the long term develop treatments for patients with that. So from sequencing the tumor bulk, we can infer part of its cellular composition, which subclones you have and what their relationships are, but there is only a limit to what extent you can do that. So when we sequence single cells from the same tumor, we can get a lot deeper in the tumor's phylogenetics and really understand what are the minute details between the cells and which events are in the same subclones and which ones aren't. So we get a lot more insight in the tumor's evolution. Our work is across any cancer types, so it's computational work in general applicable to whole genome sequencing data of cancers. We did a lot of work on breast cancers in the past, where we exposed their evolutionary history in quite a lot of detail, and a lot of work on prostate cancers as well, where we looked at metastatic prostate cancers, looked at which tumor cell populations existed, how they were related, and how the tumor spread through the body to learn about metastatic spread of cancer.